Well, maybe you could tell us a little bit about why you do think CRISPR is so great. I mean, you mentioned like the very little FDA pushback, which I'd like to come back to. I mean, part of the reason for that is because like the results are so amazing for sickle cell. So there's that, but also what's coming next? Like what are you looking at?
Yes, I do not mean to give the impression that the FDA weren't taking a very stern, long look. They absolutely did. But I think, Emma, you said it beautifully. Yes, the results were so compelling. Obviously, the scientists and the clinicians involved in the trial had been publishing in peer-reviewed journals, the New England Journal of Medicine, interim results. So the whole world could see and were pretty much unanimous in thinking and judging that these results were about as good as anybody could dream up. When the patients go into the trial, they were, by definition, very ill. They were experiencing multiple pain crises. The pain that a sickle cell patient experiences, as I'm sure you are aware of, is literally off the charts. It is absolutely excruciating, and this can result in weeks, sometimes months in hospital. And there's so little effective therapy. So as more and more patients underwent this cell therapy and came out the other side, they were experiencing a newfound energy and health that they had literally never experienced before. Pain crises disappeared. The need for blood transfusions disappeared. Hospitalizations disappeared. And this was repeated patient after patient after patient in both the Vertex trial and at the Bluebird trial as well. So 6 weeks before the ultimate FDA approval, there was a it was all open to the public, and I watched with great interest and anticipation an advisory committee meeting to really dig into one, perhaps for some, still unanswered question, which is just how safe is this therapy? You're giving patients a little RNA heat-seeking missile to go into the nucleus and identify the one stretch of the single gene or chromosome region that you want to edit, you want to cut. And the hope is that you're not somehow seeing that same sequence somewhere else in our 23 pairs of chromosomes, our 3 billion letters of DNA. But Vertex did a very compelling job of showing at that meeting, as they had for years before, there was simply no off-target effects that they had to be worried about. So while this was a theoretical concern, there was literally no practical reasons. And over the last few years, the FDA, I think, the bigger issue has really been the manufacturing of some of the viruses and some of the reagents that have been needed. That is where some of the FDA holds in some other trials have been found, not because of concerns about off-target effects. So we're all basking in the early glow of the success of the Vertex and Bluebird trials. Other companies are also developing slight variations of these approaches. We'll start to see those come up before the FDA in the next year or two, I've no doubt. And other companies are using CRISPR to attack other diseases. So liver diseases. Liver is a very spongy organ that is very good at taking up viruses and other particles. So it's a very attractive organ to target for liver-specific diseases. So a company called Intellia Pharmaceuticals has a couple of programs there. Another company up in the Boston area called Verve Therapeutics is looking at heart disease. And although they had a slight setback, their long-term goal is that gene editing could be, again, attacking the liver, could be a way to genetically reduce levels of bad cholesterol. And the number of applications is going to grow, as is the fundamental technology. So all of these systems, or most of the systems I've just very briefly summarized, have used the flavor of CRISPR for which Doudna and Charpentier won the Nobel Prize. But scientists don't stand still. And just because CRISPR has shaken up the world of gene therapy, that doesn't mean we sort of sit back and say, okay, we're done here. On the contrary, we are constantly iterating and looking for other ways to either expand on the CRISPR chassis, the CRISPR platform, or look around nature for other ways that we can kind of modulate DNA. And we're seeing amazing progress in both of those. So final point on this question, when the Nobel Committee gave out the Nobel Prize in 2020, they referred to CRISPR as the genetic scissors, which was quite nice. But the way it works is it literally cuts both strands of the double helix of DNA. Since, well, even before the Nobel Prize, other groups were starting to work on other flavors of gene editing that are much more delicate and potentially even more precise. So now we can think about surgically just fixing one letter of the DNA sequence without even having to completely cut the double helix. So new flavors of gene editing called base editing and prime editing. There are companies that are promoting these. They're in the clinic now with different diseases. So the next 5 to 10 years are going to be very exciting.